Simon Baron Cohen is a fellow at Trinity College and director of the Autism Research Centre. The very fact that the lecture theatre is full is a mark of his distinction. His title is on the screen, so I'll hand over the microphone immediately to Simon. Thank
you. Thank you very much for the invitation to talk to you today. My title is The Prenatal Sex Steroid Theory of Autism, and I thought what I would do to start is just give you the overview of the picture that I'm trying to build, which is to understand the causes of autism. It's been known for quite a long time that genetics plays a part in the cause of autism. What we've been doing here in Cambridge is looking to see whether there might be a second major piece of biology that contributes to the cause of autism, namely prenatal sex steroid hormones like testosterone and estrogen. And I'll lay out some of the evidence for that. But the idea is that these hormones might be interacting with the genetic predisposition to change brain development prenatally, and that this may be different in both males and females who are autistic. A word about autism before we get into that theory. You all know that autism is a neurodevelopmental condition affecting social skills, communication. These are children and adults who also find it challenging to respond to change, particularly unpredictable change, and may have sensory hypersensitivity. What you can see is that autism is being diagnosed much more often than it used to be. In fact, it's been estimated in a recent paper by Tamsin Ford and colleagues here in the psychiatry department in Cambridge that there's been a 700% increase in the prevalence of autism since the late 90s. So that today, the estimates are that autism is 1 in 36 children. So that's incredibly common. When I started out in this field some 40 years ago, autism was considered to be very rare, about 4 in 10,000. So there's been this massive increase in diagnosis. Probably that's to do with better awareness. It's also partly to do with changing the definition of autism to include people who have IQ in the average range, so no learning disability, whereas in the old days it was much more common that autism was diagnosed in people with learning disabilities or intellectual disabilities. What you can also see on this graph, apart from this kind of change across time, is that autism is much more common in boys than in girls, and that's really part of why we started looking at a piece of biology that might be sex-linked, such as sex steroid hormones, in terms of its etiology. So what you can see is that 1 in 25 boys now get a diagnosis of autism, and 1 in 100 girls. So the sex ratio is about 4 males to every 1 female. Whilst we're talking about the sex ratio in autism, we can also note from this study from Denmark, looking at when you get your diagnosis, it looks like girls get diagnosed later, and one relatively new idea behind this is that girls may be hiding their autism and therefore not asking for help or not seeking help. So you can see on this slide that girls in black tend to get diagnosed kind of in late childhood or with a peak in the early teens. And it could be that transition from the end of primary school into secondary school when the social demands sort of suddenly increase, that it becomes harder to hide the fact that you're struggling. But this graph also shows what we've already covered, which is that boys are diagnosed more often, and here we see getting diagnosed earlier. So the other kind of concept that's important to introduce is the idea of autistic traits. So far we've talked about autism as if it's a binary categorical diagnosis. You either have it or you don't have it, which is the case if you go to a clinic, that the clinician has to make a judgment call as to whether you meet the diagnostic criteria. But what we did, actually as part of a Part 2 project some 20 years ago, was develop a questionnaire called the Autism Spectrum Quotient, which doesn't look at whether you're autistic or not. It counts the number of autistic traits a person has. So this is a self-report questionnaire. What you can see in the general population, this is a fairly recent study published in 2018, where we were able to put this questionnaire online, and we had 600,000 people in the general population take the questionnaire, is that there's a normal distribution of autistic traits, so we all have autistic traits. Those of you who can distinguish the colours can see that in the non-autistic population, males score slightly higher than females on average. So we're seeing that link with your biological sex in terms of autistic traits, not just diagnosis. And the other two curves show that autistic people, 36,000 autistic people took part in this study online, simply score higher on average, so their distribution is shifted over to the right. So I started off saying that autism is partly genetic, and the genetic contributions to autism come from two sources. One is rare genetic variants, like mutations. They're rare because they're only seen in less than 1 in 1,000 people in the general population. And what you can see on the right of this slide is the list of rare genetic variants that have been associated with autism. Lots of genes. They're genes which are expressed in the brain prenatally. That's what you can see on the left. These genes, although they've been statistically significantly associated with autism, actually don't account for the majority of autistic cases or autistic people. In fact, they're only seen in about 5 or 10% of autistic people, but nevertheless it shows a genetic contribution to autism. The other contribution from genetics comes from common genetic variants. So these are polymorphisms, genetic polymorphisms that we all carry, but which come in different varieties. And this graph is what's called a Manhattan plot. So you've got the human chromosomes along the x-axis. The red line signifies the statistical threshold for association with a particular trait, in this case autism. And each of those dots is a single nucleotide polymorphism or a common genetic variant. The idea is that part of the genetics of autism may lie in genes that we all carry, but which come in different versions in the population, and which may occur in particular combinations in autistic people. And what you can see is, first of all, you need big data to identify common genetic variants. That's because each of those common genetic variants may only contribute a tiny fraction, have a small effect size in relation to autistic traits in this case. So you need big data to identify the signal. Here, in the largest genome-wide association study of autism to date, there were 28,000 controls and 18,000 autistic people. And you can see that just 5 SNPs or common variants cross that red line, showing statistical association. So we're seeing both rare and common genetics contributes to autism. And this kind of pie chart is another way of just visualizing what different factors account for the likelihood of autism or variance in autism. So you can see that we've talked about the smaller slices, which are the rare genetic variants. The big red slice of the pie which are the common genetic variants, but that nevertheless leaves a large slice, which is in pale blue, of non-genetic factors. So this is a long way of saying that autism is partly but not completely genetic. And the kind of demonstration of that, of course, is where you can find identical twins like these two sisters, where one is autistic and one isn't. If autism was 100% genetic, then if one had the diagnosis, the other should have too. The fact that you can find so-called discordant twin pairs, monozygotic twin pairs, suggests that there must be some non-genetic factors at play. So partly environmental factors, and this is where we have been looking at hormones, prenatal hormones, as part of the uterine environment that might be acting on the gene, so having epigenetic influences to change brain development. And in fact, in this now quite old study from King's College London, the twins study, TEDS, they did find that when you look at discordant twin pairs like this, there are differences in gene expression amongst identical twins, particularly changes in DNA methylation. And we are exploring whether prenatal hormones might be causing those sorts of changes. So before we go to the hormones, I want to kind of back up a little bit and talk about the relationship between autism and typical sex differences. And I'm going to approach this at a number of levels, starting off with the brain. So when we look at the brain, for example, in autistic people, and differences in the brains of males and females in the population, do we find an overlap? And is this driven by any shared mechanism? That's the kind of general approach. So we'll start off with evidence from neuroscience to look at this question. What you can see here is that if you do repeat MRI scanning to look at brain development, on the left we see that the autistic brain is growing faster. The brain is larger at each point. In the red group, that's the autistic group, compared to the blue group, which is typical children. So we're seeing that, at least in a subgroup, but it's shown here as a kind of group average, that the autistic brain is growing faster across the first 72 months of development. But equally, if we look at typical sex differences, so shown in right, we're looking at MRI data from the UK Biobank. This is data from the first release of structural MRI, looking at males and females. We see there is an on-average sex difference in total grey volume, total white matter volume, and total cerebral volume, with males on average having a larger volume compared to females. So one interpretation of what we're seeing on the left is that in autism, we're seeing an extreme of the typical male pattern of brain development. Now that larger brain that we're seeing in autism may translate or arise from more neurons in the autistic brain and greater connectivity in the autistic brain, because where we have the opportunity through post-mortem to look at individual neurons, you can see that a single neuron from an autistic brain at post-mortem compared to a non-autistic neuron, what you can see using the naked eye is that there are more of those white dots along the length of the neuron, and each of those white dots represents the location of a dendritic spine where the neuron is forming connection with its neighbour. So one observation is that there are both more neurons and more connections between neurons in the autistic brain. And if you divide the sample as they have in the centre here, between the younger brains, these are obviously opportunistic studies where you're looking at people who've died and divide them into younger versus older. What we're seeing in the autistic group is that there is less of a reduction in the number of connections with age, suggesting that there may be reduced apoptosis in the autistic brain. And kind of continuing this line, just looking at the number of neurons in autistic brains compared to typical male and female brains on average, you can see that, again, these old studies where they've literally counted neurons in neuropathology, is that males in the general population on average have more neurons than females, 22 billion on average in males, 19 billion on average in females. And work from Eric Corshane's lab in San Diego is showing that autistic brains have about 65% more neurons, particularly in the frontal lobe, compared to non-autistic people. So again, kind of looked at in the through the lens of how autism relates to typical sex differences, we might interpret this as autistic people having an extreme of the typical male brain. Perfect time for me to have a drink. Yeah.
Just a quick clarification. Are all of these data for autism half males, half females?
So some of the older studies, there are not that many females that are included in the studies because, it was kind of less well diagnosed in females in earlier studies. But in these studies, it combines both. You'll see in some later studies we're able to separate autistic males and autistic females. So, still looking at the brain, Meng Chuan Lai, who was a PhD student in Cambridge, used volumetric-based morphometry to do a kind of overlap study where he took autistic people and non-autistic people to look at differences in grey and white matter regions between cases and controls. And then he did the same between typical males and typical females and looked to see whether there was any overlap between what characterises the autistic brain and what characterises typical sex differences. And you can see both in grey matter on the left and white matter on the right, the autistic female brain, on average, is masculinized in showing more areas of the brain are different in autistic females that overlap with typical areas of sex difference. And Dorothea Flores published a similar sort of study just this year, looking at an even larger cohort of brains using MRI. So using the UK Biobank, she had 10,000 typical brains, and then in 2 different data sets was able to look at about 2,000 autistic brains and employed machine learning to classify sex differences in the typical brains and see how well the autistic brains could be classified as male or female based using these algorithms. And what she found was that brain structure associated with males is more predictive of autistic males, and brain structure associated with females is less predictive of autistic females. And so she concluded that we see a male shifted neurophenotype in autism. And she went on to look at functional MRI as well. So looking at the default mode network or resting state, which, as you know, is the pattern of brain connectivity that you see, or brain activity that you see when you're not doing anything, but which is associated with theory of mind, because often we're reflecting on our own or other people's mental states. What she did again was to look at whether there are sex differences in the default mode network, or the DMN, which she found that there were, and then looked to see whether the pattern of sex differences was different in autistic brains compared to non-autistic brains and found, again, just in autistic males, that there was a shift towards increased patterns of maleness in the autistic brain. So some evidence for relationships between how the brain is developing and the structure and function of the autistic brain and typical sex differences. What about when we move to the level of psychology? Our group has been quite interested in, first of all, empathy when it comes to typical sex differences in the mind, and also how this relates to autism. We developed this test called the Reading the Mind in the Eyes test, sometimes referred to as an advanced theory of mind test. So you look at photographs of just the eye region of the face, and you have to say which of those 4 words best describes what the person in the photo is thinking or feeling. So it's a test of empathy, cognitive empathy. What's found in many studies is, again, a sex difference or gender difference on average. So you can see at the top there that when we've had the chance to put that test online and get large numbers of people taking the test, females in green, top left, on average score higher than males in blue. And if we look at the pattern of scores in autistic people, so the bottom left-hand panel, you see again the female advantage in empathy compared to typical males. And autistic males and females, the 2 left-hand bars, they score on average lower than typical individuals. But again, their pattern of results suggests that in autism we're seeing an extreme of the typical male profile. And when we ask people to take that test whilst they're having an MRI scan, so cognitive activation, what you find is, again, a sex difference in the typical case with increased activity in the left inferior frontal gyrus whilst they're looking at these photos of the face to decode facial expression. Females showing more activity in that region than males on average, and autistic people showing underactivity in that same brain region whilst they're doing the test. That sex difference that I just mentioned on the eyes test, we recently replicated that in a very large data set. It's the kind of beauty of now being able to do online cognitive testing. 300,000 people took part across 57 different countries. So we published this in PNAS this year. And you can see that in different cohorts we were able to replicate the finding that females on average score higher than males on this test of empathy. And you can see that there are all the different countries of participants that took part, and the sex difference is apparent across age as well. So lots of big data supporting the idea of a sex difference in empathy, and in autism, people struggling with cognitive empathy. And a second way to measure empathy is through questionnaires. So we use something called the empathy quotient, where you kind of report, you self-report how easily you can put yourself into someone else's shoes to imagine what they're thinking and feeling across a number of items. Again, big data, we were able to test 600,000 people in the general population, 36,000 autistic people. You see very clearly that typical females in the population report having better empathy scores compared to typical males, and autistic males and females being shifted over to the left on this empathy test. So again, sort of fitting this idea of the autistic mind being an extreme of the typical male mind, at least in relation to this dimension of empathy. And another kind of side to cognition we've been looking at is attention to detail, and building on really old work using this test called the embedded figures test, where you have to find the target shape as quickly as you can in the overall design. I won't trace it out for you, but some of you are looking at it right now. And what's been found over half a century of research is that on average males are faster than females at finding the target within the complex design. So the numbers there reflect the average number of seconds it takes to find the target shape, males being faster on average, autistic people being even faster and more accurate than typical males. So now we're seeing, the same pattern, which is the autistic mind showing an extreme of the typical male mind, but this time showing superior performance in autism. And when you ask people to take that test whilst they're having an MRI scan, we find that non-autistic males show less activity in the posterior parietal cortex whilst they're doing the task compared to typical females. So better performance is associated with reduced activity in this part of the visual cortex, and autistic people show even less activity in that same brain region whilst they're doing the task. Attention to detail, I think, is part of what I call systemizing, which is the ability and the interest to analyze a system in terms of its rules, where it pays to pay attention to the detail of the system. Here we're using the systemizing quotient, another questionnaire, where we ask people how interested they are in different kinds of systems, whether they're mechanical systems, natural systems, mathematical systems, etc. And what you see is in the general population, again, big data, males on average report stronger interest in systems compared to females, and autistic males and females report even higher scores on this systemizing quotient. So one thing that we can do is put together these different measures by asking people in the population to take both the EQ for empathy, the SQ for systemizing, and plotting where individuals lie in this space. So each dot is a person who's taken the EQ or the SQ. What you can see just visualizing it is that more women than men in the population, that's in yellow, score in the region where their empathy is at a higher level than their systemizing. More men in the population than women, so men are shown in green, score in the range where their systemizing is at a higher level than their empathy, and autistic people shown in purple and pink, males and females, are scoring down in this bottom right-hand quadrant, an extreme of the typical male profile, where their systemizing is anywhere from intact to above average, whilst their empathy is below average. And you can see, apart from just looking at it with the naked eye, that you see more males showing this profile of type S, systemizing higher than empathy, more females in the population showing type E, empathy higher than systemizing, and the majority of autistic people showing either type S or an extreme of type S. So kind of clear patterns suggesting a relationship between how autistic minds and brains develop and function and typical sex differences. And this just shows you what we've already mentioned, which is the D score, the difference score between your empathy and your systemizing, is shifted over in autism in a masculinized direction. So where do all these sex differences come from? And now we can start to kind of track backwards to biology. Because obviously a lot of sex differences or gender differences that we've been talking about so far could just be the result of postnatal experience and socialization and culture. In order to test this, we carried out a study at the local maternity hospital here in Cambridge some 20 years ago, where we tested newborn babies aged 24 hours old who were presented with either a social stimulus, which is a human face, or a non-social stimulus, this geometric design. And we filmed the babies. Two master's students did this to see how long they looked at a social versus a non-social stimulus. What we found, just cutting to the chase, is that more girls than boys looked longer at the social stimulus, the human face, and more boys than girls looked longer at the non-social geometric design. Obviously there were babies who looked equally long at both, but here I'm just plotting babies who showed a difference in their scores. The fact that we find any difference at all at birth, or as close to birth as we can get, suggests that whatever the role of culture in shaping sex or gender differences in the mind and the brain, prenatal biology must also be playing a role. And actually looking at data from the Developing Connectome project from King's College London, where there's now MRI data from newborn babies, we can see evidence of sex differences even in the brain at birth. In this case, we're looking at females showing greater functional connectivity in visual association areas. So some of the sex differences we see in the typical brain must be partly to do with prenatal biology. Coming back to very young autistic kids, at the earliest stage that you can diagnose autism, which is at about 18 months to 2 years old, this study, again from San Diego, showed that if you present the social stimulus and the non-social stimulus and record looking time at each, if a child looks for more than 70% of the time at the non-social stimulus, the probability that they're autistic is 100%. So this is kind of a striking behavioural test, very early in development. We don't know if we'd get the same levels of accuracy if this was done in the general population. This was obviously a clinic sample, but looked at side by side with that newborn baby study, where we know that there's a typical sex difference in how attention is allocated to the social and the non-social environment. Again, what we can say is at the earliest point in development, autistic kids are showing an extreme of the typical male pattern of attention. So let's sort of move now to the sex steroid hormones. So the sex steroids include testosterone and estrogen. What I'm going to do in the next part of the talk is just run through some studies which have shown differences in circulating levels of these hormones, which are obviously much easier to measure because you can ask people to spit in a tube, give a blood sample or some other biological measure, but it's from people who are able to consent. So this study, which we conducted now more than 10 years ago, came from blood samples, serum samples. What we found is if you look at the sex steroid pathway, this is called the delta-4 sex steroid pathway, which starts with progesterone and goes through a series of steps, each androgen being synthesized into the next through the action of a hormone, of an enzyme. Each of these are hormones, and they all have masculinizing properties on the body and the brain. What we found just looking at serum samples was an elevated level of androstenedione, so it's one step before the synthesis of testosterone in both males and females with autism, suggesting dysregulation in the hormone system. This study from Vanderbilt University in the US collected salivary testosterone, waking salivary testosterone because the hormone varies diurnally, but looking at it across age and finding higher levels of testosterone in autistic males compared to typical males across development. So suggesting that there is some hormonal dysregulation in autism. This study from Switzerland looked at, again, a range of different steroid hormones. In this case, they were looking at androstenedione, finding increased hormone androstenedione in autistic boys compared to controls who are matched for age, weight and height. And in this study from Egypt, they looked at a number of different hormones that are related to the sex steroids, so free testosterone, DHEA, and delta-4 androstenedione. So in each case, the autistic group showing higher levels than the non-autistic group. And this has been replicated in 2020 in a Swiss sample. In this study from Poland, they looked at adrenal steroids, because the steroids can come from either the adrenal glands or the testes, amongst other sources. And again, comparing autistic males and females with typical males and females, but looking across age, they found a different pattern in the rate or the levels of hormone levels, the levels of hormones across age in the autistic group, with elevation in adrenal steroids in the autistic group. And this was measured in saliva. And a final study, I think, which comes from the States, from Drexel University, looked at a high likelihood autism sample. So this is where you can study the babies of in families where there's already an autistic child, so pre any possible diagnosis, but where these babies for genetic reasons may be at a higher likelihood of developing autism. And they looked at the meconium, which is the first stool produced by the baby, as a reflection of prenatal androgens, and found that there was a positive correlation with a measure of autistic traits, the SRS, particularly in boys. So some suggestion that autistic traits in the population in this particular cohort enriched for autistic traits for genetic reasons is associated with prenatal androgen levels. So to get at kind of prenatal hormones, because we know that autism starts prenatally, to the extent that these hormones may be playing any role, we need to measure them at the critical period in development. This is much more tricky, and this is what we've devoted quite a few years to doing, and I'll tell you how we've been doing it. First of all, this just shows you a schematic that if this is birth, and these are the weeks before birth, you can see that prenatally there's a surge in the production of testosterone in both boys and girls, but particularly in boys, shown in blue. There's a second surge postnatally around 3 months of age, and then a third one at puberty. We know what the puberty surge of testosterone is related to, which is secondary sexual characteristics. But there's been a big question about whether the prenatal surge in testosterone has a different role, which is to do with masculinization of the brain and the body. What you can see here is that surge in testosterone between about 12 and 20 weeks of gestation coincides with when amniocentesis takes place. Amniocentesis is a clinical procedure where some women have fluid, the amniotic fluid, sampled from around the baby. So this gives us an opportunity to measure prenatal sex steroid hormones like testosterone and estrogen at the time in development where it may be having an organizational effect on brain development. So I've used that word, organizational effects. It actually comes from the term organizational effects comes from a neurologist called Norman Geschwind working in Boston in the 1980s. He was arguing that prenatal hormones like testosterone and estrogen permanently change the structure of the brain. Boys produce more of these hormones than girls because it comes from the testes. You can see the pathway. I've already mentioned the delta-4 pathway from progesterone through to androstenedione and testosterone. And testosterone is converted into estrogen at the final step there through the action of an enzyme called aromatase. We've been measuring it in women who have amniocentesis during pregnancy. It's the closest we can get to the fetus in an ethical way. And this is research that's piggybacking on the fact that these women are having the clinical procedure anyway. It wouldn't be ethical to ask women to do this for research, but if they're doing it anyway for clinical reasons, we simply ask them for their consent to analyse the fluid, which would otherwise be thrown away. In Cambridge, we've been following a cohort of children whose mothers had amniocentesis, and when the children were 8 years old, we called them in for an MRI scan. It's the first time in the case of humans that we were able to demonstrate correlations between grey matter volume in different brain regions and prenatal sex steroids. So you can see a number of different regions show a correlation, including, for example, the STS, superior temporal sulcus, which is associated with face processing and social perception, the planum temporale, which is a language area. And so this was certainly showing that once you can measure prenatal hormones like testosterone and then look at later brain development, the hormones seem to be associated with, arguably causing, changes in brain structure in humans, something that we'd known for a long time from animal research, but had never been shown in humans before. When these kids were 8 years old, we also asked them to take that test of empathy. So this is a child version of the empathy test, and found that the higher your prenatal testosterone, the more difficulties you're having at reading faces to decode facial expressions at 8 years old. Here we've got a few hundred kids taking part at most, and girls are shown in red, boys are shown in blue, and you find these effects within a gender as well as when you combine genders, suggesting it's a hormone effect rather than a sex difference. At 8 years old, we also asked the kids to take that test of attention to detail, the embedded figures test. So now you have to find that triangle as quickly as you can in the overall design. And now we're finding a positive association. The higher your prenatal testosterone levels, the faster and more accurate you are in finding the part within the whole. So the same hormone seems to be having different patterns of association with either social or non-social cognition. We asked the mothers to fill in the autism spectrum quotient as a way of counting how many autistic traits the children have. What you can see is a positive association. The higher the prenatal testosterone, the more autistic traits the child is showing at 4 years old. I should say that this is something like a double-blind experiment, in that the mother has no idea what her child's prenatal hormone levels were. The biochemist measuring the hormone levels has no idea how the child's autistic trait measures are going to turn out 4 years later, and then you put the data together to see if you find an association which is apparent here. That's to do with children here in Cambridge who were part of a kind of normative or typical cohort. They were only being studied because their mothers had amniocentesis. But to really test the hypothesis about whether prenatal sex steroid hormones play a role in autism, we would need much larger samples. We said earlier that autism is 1 in 36 kids, so about 3% of the population. And in our studies in Cambridge, we only had about 200 kids in total. So we needed much larger samples to test the hypothesis. So we collaborated with the Danish Biobank, who have been collecting amniotic fluid since the 1980s, very conveniently, and where we could link the amniotic fluid in the biobank with the Danish psychiatric register. That's to say a population register that documents who in the population has been given a diagnosis, in this case of autism. On that basis, and just focusing on children born in the 1990s, where autism diagnosis would be kind of equivalent to what it is today, we were able to find 128 autistic kids whose amniotic fluid was in the deep freeze, and as many controls as we liked. And what we found was that we only looked at this in boys. There weren't enough autistic girls to look at. Is that autistic boys had a higher level of prenatal sex steroids across that whole delta 4 pathway compared to typical males, who in turn had higher level compared to typical females. And then in a few years later, we were able to look at the role of prenatal estrogens, not just androgens. And it turns out that the prenatal estrogens seem to play an even bigger role in relation to autism. The odds ratio is larger than the androgens, but certainly confirming that in autism, prenatal sex steroids are involved. So this begs the question, where is this elevation of the prenatal sex steroids coming from? And there are three possible sources. The mother, she obviously has her own hormones that could cross the placenta, and the baby could then be exposed to higher levels. The baby, who is synthesizing his or her own hormones. And then a third possible source is the placenta, which synthesizes its own sex steroid hormones. And some of our research is now kind of looking at each of these sources to try and isolate where is the elevation coming from. But certainly there's some evidence for the mother's hormones being part of the answer, because in this study from China, published in 2015, you can see that mothers of autistic children who gave a blood sample during pregnancy had higher levels of testosterone compared to mothers of typically developing children. And this has been kind of confirmed in a more recent study just this year, looking at so pregnancies that result in autism, they call it ASD, and pregnancies that don't result in autism. And they looked at an extra factor, which is whether the mother had what's called prenatal metabolic syndrome, which includes things like gestational diabetes and preeclampsia, which can also change the hormone environment. But what they found was that that pregnancies associated with later autism had higher levels of estradiol, one of the estrogens, compared to pregnancies that did not result in autism, independent of whether the mother had this prenatal metabolic syndrome. And in a kind of final test of this, we had a master's student who looked at mothers who have the condition of polycystic ovaries syndrome. So polycystic ovaries syndrome is in about 12% of women in the population. It's characterized by irregular menstrual cycles, late onset of puberty, and excess bodily hair. It's driven by high levels of testosterone. So these are women who have high levels of testosterone. What we found is that women who have polycystic ovaries syndrome have a higher likelihood of having an autistic child. We found that in the UK, and it's subsequently been found in 3 other countries, Finland, Israel and Sweden, suggesting that the prenatal uterine environment in which the baby finds itself is changing brain development because of hormonal exposure to increase the likelihood of autism. How do prenatal sex steroids work? Just in the last 5 minutes, the mechanism of action, there seems to be a consensus with this kind of cartoon, so it's still kind of valid, which is that hormones like testosterone, the molecule can cross into the cytoplasm of the cell. As I mentioned earlier, it can be converted into estrogen through the action of the enzyme aromatase. But whether it's testosterone, an androgen, or estrogen, it has to bind to a receptor, an androgen receptor or an estrogen receptor, and the receptor can then enter the nucleus of the cell and bind to the DNA itself. So the hormones are effectively operating as a transcription factor and changing gene expression. And according to this kind of view, one of the consequences is changing the rate of apoptosis or selective pruning or selective cell death. A test of this came from Madeleine Lancaster's lab here in Cambridge. She works at the MRC LMB, Laboratory for Molecular Biology. What she's been doing is adding androgens to organoids or mini brains so that in a dish you can look at the effects of extra androgens on brain development in an ethical way. And what you can see is that when you add testosterone, it not only increases the number of neurons, we saw that earlier in relation to autism, it also increases the number of symmetric proliferative divisions and an increase in the number of excitatory neurons. So this is giving us a kind of model system in which to look at how do early prenatal androgens affect brain structure and brain function. And people are also doing similar work in animals. So in this study of rats, you can give female rats extra testosterone to see what happens to her offspring. And what you can see is that in the offspring of rats who were given extra testosterone, in this behavioural test of do they choose to spend more time with a conspecific, so a social situation, or an empty cage, a non-social situation, you see that extra testosterone reduces the amount of time spent with a conspecific. And when you look at what's changed in the animal's brain following exposure to extra testosterone, you can see in the female rat increased in the number of neurons and neuronal connections, particularly in the hippocampus. They also found a decrease in a different hormone, oxytocin, in the rats whose brains had been exposed to extra prenatal testosterone. So all of this is adding up to a new way of looking at autism. This is kind of drawing things to a close, and then we can have time for questions and discussion. But the idea that development prenatally is shown along the bottom here, across pregnancy, the first trimester, second trimester, third trimester, and that in autism we see the influence of genetics up at the top left. We see genetics interacting with these hormones which start to surge in prenatal development, particularly estrogens, particularly around the second trimester, affecting synaptogenesis and neurogenesis. Quite what the role is of the placenta is the subject of our current research to see whether that's also contributing to this change in hormonal environment in autism to change outcomes. So we return to this kind of overview that I hope I've laid out some evidence that in autism there's an interaction between prenatal sex steroids and genetics to influence prenatal development in autistic males and females. And I'll stop there, invite you to visit our website if you want any more information about these studies, and thank our funders and the big team who do the work. Thank you.
you for a talk of exquisite clarity and elegant construction and perfectly matched to the Samuel Club audience. Thank you very much indeed. I'm going to hand over the questions to Simon in a moment, but traditionally at the Samuel Club we now have a moment's break where those who have to go off for their various business can slip away, and those who want to listen to questions can stay. So we'll have a short break.
Good.
That was lovely.
Good.
And thank you for the impeccable time. You're a real professional.
Right.
I guess they're mostly Part 2 students. But do you recognise a lot of people?
I recognise some of the ones from Trinity because I'm a director of studies. Although we've got Richard Bethlehem, who's just been appointed as the second director of studies.
Yes, you're fortunate.
So that's good.
So you said it gets a bit stuffy. No,
Here.
what were you going to say?
It is a little bit in here, but that's okay.
Oh, we can open the window.
Right.
Shall I open the window?
Is there a way of controlling it? I think it's fine. No one's complaining.
So questions from Professor Baron Cohen?
Yeah. Do you want to go first? Right.
Some of the sample sizes that you spoke about were really impressive, and I'm just wondering if they were entirely westernised or if they were representative of like socioeconomic groups or ethnic subgroups. And then I guess a sort of part B to that is if they were westernised, were there any other like cross-cultural replications of the work? Because I feel like that can be quite helpful in understanding the role of socialisation.
Yeah, I mean, I think you're right that that culture must be playing a part in a lot of this. So there are no strong claims being made here. You know, that the mind is the result of our experience, which varies culturally, but also partly our biology. In answer to your question, I think there probably is a bias towards research being done in Western cultures, just as a reflection of where does science take place. I think in some studies I was able to kind of show some of the cultural variation, and there was that particular study looking at the eyes test across 57 countries, which did include, I think, some non-Western ones, but I'd have to look back. But I suppose when it comes to prenatal biology, in a way you can kind of set culture aside, because what you're looking at is events that are happening before culture has had much chance to operate on the human mind. Yeah. Yeah.
I have a question really about kind of, I guess, the attribution of all these parallels between sex and autism-related differences, because historically autism's kind of been constructed as a diagnostic category coming very much from male populations, and whether this kind of body research reflects actually more that it's a hyper-masculinized autism profile that we're still working with, and therefore that's why it's kind of people present more similarly to males, rather than if it's reflective like properties of autism itself.
Yeah. Okay. So I think this sort of relates to the question that I got early on, to do with so you know, in these studies, how often are autistic females included compared to just autistic males? But I think you're also asking, you know, if there's been this kind of bias towards studying autistic males for the longest time, does that mean our kind of way that we even look at autism has been influenced by some kind of bias?
The autistic females are more likely to then be a more masculinized profile because they're the ones who've been picked up within that model.
Yeah.
I'm just interested if there's been kind of, also any like research into kind of probing what biases may be present in different mechanisms.
Yeah, no, I mean, I think it's a good question, and I think as more females are being diagnosed as autistic, well, one question is that going to change the way we diagnose autism? You know, just kind of looking at different sets of so-called symptoms or signs. and will it still kind of show the same patterns in terms of hypermasculinization? I think it's kind of early days, that although we've seen this increase in the number of autistic females getting diagnosed, in terms of them also taking part in research, it's still quite recent. But it's a very good point. Yeah.
I'm not sure if this is a silly question, but obviously when people have really severe autism, that can affect their well-being. But are there any evolutionary benefits to the increased masculinization of the brain and therefore autism?
Right. I mean, evolutionary accounts are kind of quite difficult to test. We can speculate, because you could argue that if you've got a disability that affects social skills and communication, that might affect your reproductive success. But on the other hand, autism isn't just defined by those disabilities. Autism is also it involves differences. A lot of people are kind of talking about neurodiversity, that autism is one example of differences in brain structure and function and different profiles of strengths and weaknesses. And we saw in some of those studies that autism confers a benefit when it comes to attention to detail and interest in systems. You could speculate that that could have conferred evolutionary benefits, that people who are better at understanding systems might have been better at developing new methods, new tools, for different kinds of resources. But as I say, the kind of evolutionary approach is hard to test. Do you want to come in on this?
Yeah. Is it known yet whether autistic people disproportionately survive COVID? I mean, one of the great pressures on mankind has always been great epidemics, and at times in our history, large proportions of the population were taken out by epidemics.
Right.
And you catch diseases from other people,
Mm.
and autistic people might be expected to survive because they don't interact closely with other people.
Right.
So I've been wondering whether there was a disproportionate survival of COVID.
Sadly, we've got some data suggesting the opposite. So we've just submitted a paper using NHS data, looking at what happened to autistic people during the pandemic, and finding not only higher rates of hospitalisation, but higher rates of mortality during the pandemic. So, I mean, this is kind of still to go through peer review, so it's unpublished, but that might fit with other findings that autistic people tend to have tend to have all sorts of physical health challenges. It's not quite clear yet why, whether that's to do with lifestyle, genetic predisposition, hormonal differences. So it doesn't quite fit your prediction.
No, I was wondering whether there was evidence, and that would be clear evidence against it.
Yeah. So, go ahead.
So the 700% increase in autism diagnosis in 2019 is quite amazing, and I'm just wondering how you, that leads to variability in how we understand autism, and then when you're trying to look for effects in genetics and hormones, more noise. So how are you dealing with that going forward?
How am I feeling?
Dealing with it.
How are we dealing with it?
To that increase?
Right. So I suppose one way that you could deal with it is to kind of look at different cohorts across time. So if you're testing particular hypotheses, you can look to see, do they hold true in different cohorts across time. I think the nature of what we call autism is changing. So if you went back to kind of pre-1990s, the profile of the population was very different. 3/4 of autistic people also had learning disabilities, whereas today it's the mirror opposite. 3/4 of autistic people do not have learning disabilities. So it just means that what we call autism has shifted quite significantly. I think you're right that changes genetics, our understanding of the genetics, because some of those rare genetic variants that I talked about earlier are much more often associated with autism with learning disabilities. They have a big impact on things like language delay and developmental delay. So yeah, I think probably the way that we need to deal with it is through these kind of cohort approaches, just because we may not be otherwise comparing like with like. Yeah, Barry.
Can I go into the neuroendocrine bit of this? Because what you're describing, organisational effects of steroids on the brain, sexually dimorphic development of the brain, that comes from a slew of research in the 60s and 70s, which showed that if you deprived males of the equivalent stage of pregnancy as postnatal in rodents, the first few days after life, it deprived them of androgens, let them grow up. They show female patterns of sexually dimorphic behaviour, for example, in the tested population, and vice versa. If you expose female brains to androgens, which acts by conversion to estrogen in this case, they show masculine patterns of behaviour, sexually dimorphic behaviour in adulthood. So I'm just wondering, and those effects are often mediated by the effects of steroids on the subcortical brain, so it's the preoptic area, for example, sexually dimorphic, not the cortical brain, which you tend to be talking about in terms of cognition. So what I'm wondering is, first of all, are any of these effects mediated by those bits of the brain that actually have steroid receptors, which are not cortical but subcortical? And when you talk about boys and girls and sex, is that what you mean in terms of their behaviour, or is sex in any way discordant with gender?
Right.
And the early work of John Money and Anke Earhart, which agree, pointed out that discordance between humans who are exposed or not to androgens in utero or on the spectrum showed more or less levels of concordance between their behaviour and their gender identity.
Right. Thank you for that set of questions.
Racking my brain from an earlier life.
Yeah, no, it's great. So some of those early studies that you talk about from like the 1960s and 70s, from animal work mostly, they were mostly kind of looking at things like mating behaviour. So it was kind of a narrow slice of animal behaviour, whereas we're looking at, aspects of behaviour and cognition which is much broader than that, like empathy and attention to detail and it is, or social interest, which you can look at in rats. But your question about sort of, was it just in the the preoptic area subcortically? I think both androgen and estrogen receptors are found widespread across many brain regions, so subcortical and cortical. I think with PET. We may be able to start mapping the distribution of these receptors in the human brain, and that's what we're hoping to do both with typical males and females and with autistic people. So kind of using PET, where you can have a ligand that binds to the receptor, so you can sort of see where are the hormones actually acting in the brain. And then there's this puzzle that amongst autistic people there's an elevated rate of transgender. And I think this might go to the second part of your question. Both ways, actually. I think female to male is the more common form of transgender. But with Varun Warrior, who's now in the department, we found that autistic people, I think, were about 6 times more likely to be transgender, so not identifying with the gender that they were assigned at birth. And we wonder whether that might have, a link to the prenatal sex steroids or differences. But of course that could be determined by lots of different factors. It's something else that needs to be explained, why autistic people are not identifying with their biological sex as much as non-autistic people. Yeah.
Thank you very much for the amazing talk. So currently the treatment for autism is is largely based on behavioral intervention and
Mm-hmm.
psychoeducational intervention.
Yeah.
Applied behavioral analysis, explicitly teaching them social skills. So I wonder, based on the findings on a hormonal level, does it inform other intervention strategies to treat or even prevent autism from
Right.
a biological perspective?
Yeah. So I don't know whether you intended to do this, but this is quite a controversial question. So it's controversial for two reasons. One is the idea of a hormone treatment, and we can talk about that for a second. And the other is the idea of preventing autism. So in terms of the first of those, the treatment idea, from the work I've been talking about, if hormones are elevated prenatally, sex steroid hormones are elevated prenatally, it might kind of give rise to this idea that perhaps you could block the hormones, and would that change the pattern or the path of development in terms of the brain and the mind. But obviously these hormones might be involved in many different aspects of the body and the brain. So if you were manipulating the hormones, you might change autistic traits, you might change empathy, you might change attention to detail. There's a lot of different, you might change your gender identity and your sexual orientation and all kinds of things. So these hormones have widespread effects in the body, so one would want to tread carefully. Second reason why it's controversial is that amongst many autistic people, they're not actually looking for a treatment. They're saying we are different, so this is part of neurodiversity. We want support for our disabilities, but we're not necessarily looking for treatment for our autism itself, because autism is just part of the variation in the population. So somehow we have to find the right balance there. And then the third part, which I think maybe you kind of wandered into this territory without fully, appreciating the context, is that the idea of preventing autism. There's a long history in the field of disability of prevention of people with disabilities. The clearest example being Down syndrome, where a lot of women, when they're pregnant, might opt to have a test to see whether their fetus has Down syndrome, and on the basis of the results of the test, choose to terminate the pregnancy. So they're kind of preventing the disability in the most extreme way, which is termination. And that obviously has big implications for how we think about disability in our society, and kind of the message we send out about disability. In fact, it's been argued that something like 90% of people with Down syndrome who would ever have lived have been prevented from living. So that's how successful the kind of prevention and prenatal screening has been for certain disabilities. But autistic people, quite understandably, are saying, is research like this, is there a risk that it could lead to the prevention of autistic people? And if you took any other minority in the population, obviously they would feel very kind of threatened by the idea that they might be prevented. And I think we need to extend the same kind of sort of thinking to autistic people. So it's kind of a long answer, but just to say that there's lots of kind of bioethical issues behind the idea of treatment. Certainly, I think if symptoms are causing distress, then they need treatment and prevention, but those are to do with symptoms rather than autism itself. Yeah.
I was wondering if your hypothesis is that it's probably the mother hormone levels that impact on the development. How would you explain the case of the twins? Right?
Yeah, right. So I think what I was trying to sort of say at the end of the talk was that the mothers may be part of the story. The fetus may also be one of the sources of elevated hormones, and then the placenta is a third source. And the kind of one possibility is that all three components are interacting. But you're asking about identical twins, is that right?
Yeah, because at the beginning of the talk you mentioned the case of
Yeah.
identical twins.
Yeah. And identical twins sound straightforward, but it turns out they come in different varieties, because some identical twins share the same amniotic sac, so they should be exposed to the same level of hormones, but some identical twins actually have their own sac. Some identical twins share the same placenta, so if the hormones were coming from the placenta, they should be equally exposed, but actually some identical twins have different placentas. So it kind of the story is a bit more complicated, and it may be that where you have discordant identical twins, one is autistic and one isn't, that they may not have either shared the same sac or the same placenta. And I think a last complication is that hormones kind of diffuse along a gradient, and so it could be that the position of each twin also influences their exposure to the concentration of hormones. So lots to unpack. Yumna.
So I have a two-part question
Uh-huh.
on the first half of your talk rather than on sex steroids. So you presented these statistics where autism is more common in males than females. So a very common counter-argument to this is the idea of masking in females. So do you not think that the very idea of masking in females is actually evidence for sex differences?
Do you mean that if autistic girls are masking more than autistic boys are, that it's by itself that is a sex difference? For example, maybe they've got better skills at hiding their autism, because it takes, maybe some social skills to be able to imitate a neurotypical pattern of behaviour. It's a very interesting idea. I think masking has mostly been looked at as a negative thing, that people are maybe experiencing stigma in relation to their autism, so they're trying to hide it. But at a certain point it just becomes too difficult to hide it, maybe because it's causing too much stress and anxiety. So the mask kind of slips. But I think your kind of quite novel interpretation of it is that masking itself may be a sign of good imitation skills, for example, and that might come more easily to autistic females. It's probably testable. I haven't thought about how. Yeah.
Just follow up on this question.
Yeah.
I've just been wondering whether systemizing can actually be used as an explanation for masking. So obviously we find evidence that autistic people score higher on systemizing traits.
Yeah.
And the concept of systemizing is based on if-then rules.
Yeah.
So do you think that this advantage can actually extend to social rules? So you use these if-then rules to kind of learn social rules, which in turn makes you apparently better at empathizing, but instead it's actually just masking.
Yeah. If you talk to autistic people, often they describe trying to systemize the social world. And if you talk to non-autistic people, that's not usually the approach that they take to conversation, for example. You don't go into a conversation or into a relationship trying to identify the rules. It's much more intuitive than that. So it could be that, what autistic people are doing, even when it comes to social skills, is trying to systemize. It hasn't really been looked at yet, but I guess what you might expect is that if you took a systemizing approach to social interaction, it could work to a certain level, but it may not work as successfully as a more, I'm going to use that word intuitive, because it's not a very good kind of account of how non-autistic people succeed in socializing, but we've seen that, some of socializing includes things like theory of mind or cognitive empathy, which is not really rule-based. But I think this is probably open to being studied. Yeah.
How
how do you reconcile the earlier idea of autism traits and the autism quotient with this new sort of theory of steroids hormones? So would it be that there's a certain threshold of having more testosterone, prenatal testosterone, that would sort of in the binary way make you more likely to be diagnosed later, or do you think it's more of a continuum where certain levels of hormones prenatally will then map onto certain levels of autistic traits? Like,
yeah. So obviously hormones exist on a continuum, and so do autistic traits. And what we've seen across a number of studies is that there's a positive correlation between those two. So the higher your prenatal hormones, the higher the greater the number of autistic traits you have. But autistic traits alone is not what determines whether you need a diagnosis. You get a diagnosis if you're struggling, basically. So you could have a high number of autistic traits and not need a diagnosis. Perhaps because of masking, or because you found yourself in an environment where you're accepted and you can just be yourself, and so you're not unhappy. So the kind of relationship between autistic traits and diagnosis, I think itself is quite, it needs more research, because something must have to tip you from having a high number of autistic traits into the experience of not being able to cope. And that seems to kind of vary a lot. It may be to do with how much support you get or whether you feel you belong, but I think it needs a lot more research. But certainly this idea that, you know, prenatal hormones may be playing a role in how many autistic traits you have, I think that's kind of consistent with the concept of a spectrum.
And sorry, just
yeah.
Up on this, would those traits, would some of those traits be more dependent on hormones than others, or is it just completely linear?
Well, actually, no, that's a really interesting question, because when you look at autistic traits, some of them are social, like how interested are you to socialize, how much, you know, how strong is your drive to socialize. Some of the autistic traits are non-social, like whether you have a good memory for phone numbers or strings of digits or other kinds of patterned information. And what I find interesting is that the same hormones seem to be involved in those very different autistic traits, but in opposite directions. So positively associated with attention to detail and systemizing, and then negatively associated with the social ones. So it's almost like there might be a trade-off, that as your hormone levels go up, you're going to get more of some kind of traits and less of the other, that the social and the non-social aspects of autism are not completely independent. We've got time for a few more. Yeah.
Yes, thank you for the insightful presentation. Yes, I'm also asking regards to traits. You realise you have a number of traits. Now, how much do we know how the symptoms of the traits fluctuates through your environment or your mental situation? And so, you know, if you're happy, if your situation is comfortable, will your traits display less? Or in adolescence, also because of hormonal changes, is there likelihood of traits to rise?
Right.
And do we know much about that?
We don't. I mean, it's a great question. So I suppose we could ask people to kind of take the test of autistic traits multiple times, I don't know, a day or over time, to look at fluctuations. I think the assumption is that it's pretty stable. So if you score high as a child, you'll still be scoring high as a teenager or as an adult. But it's an open question. I don't think people have looked, actually. So there's this assumption that because it's a trait, it's a stable characteristic. But it may be that it fluctuates. That's very interesting. And as I say, the simplest test of that would just be test-retest scores, you know, even across the same day.
Yes, and then there's a question of then a treatment pathway if there is a need because traits of race or something.
Yeah. So, I mean, it goes back to the earlier question about treatment we talked about earlier. But I think if someone is looking for support or intervention or treatment, it's you have to first of all, focus on which aspect. So, which symptom, for example, is it that they want treatment for? And I think you're asking, might that fluctuate for all kinds of reasons at different points in their development? But certainly, symptoms like anxiety, which are very common in autistic people are often asking for treatments for anxiety. And we don't really know whether they're, for example, pharmacological treatments for anxiety that work in non-autistic people are as effective in autistic people. At the back. Yeah.
I was wondering about what is the reception of theory among autistic people and their families. For example, it depends how we explain it, I suppose. Like, how would you explain it? For example, historically, some findings of genetic phases of autism have been associated with, I think, some destigmatizing of autism because it removes the focus from more psychological theories,
Yeah.
such as mother refrigerator theory. So,
Yeah.
what's the best way to explain this theory to families?
Yeah. So, I think the genetic theory has now been kind of accepted. Doesn't mean that there's not some resistance to it, because we study genetics of autism here in Cambridge. I know you do too. And some autistic people worry about how is genetic research going to be used. So although it's moved things away from blaming the mothers for causing their autism, so it's kind of moved it into biology, there's the kind of a new fear which is, why are you collecting our DNA and how could this be used and could it be used to harm us? So I think we need to be kind of always talking to the autistic community to include them in the design of studies and to ensure that they feel trust in the research. If you're asking about the prenatal sex steroids, I think some of it we would explain to autistic people and to families in terms of just trying to understand the causes of something, and that is kind of what science does. But here we can also sometimes see it has medical relevance. So, for example, discovering that autistic women are more likely to have polycystic ovary syndrome, which is a hormonal condition, that came out of the research looking at prenatal sex steroid hormones. So, it might help make sense of some of the medical profile of autistic people because these hormones are not just affecting brain development, they could be affecting the whole body. I think it's 1:30, so we'll just take one last question if there is one before we stop. Yeah.
Can I ask you about the sample for which, obviously you said for ethical reasons, obviously you're using amniocentesis, but
Yeah.
often the women who go on to have amniocentesis are older,
Yeah.
or they have already detected certain anomalies in
Yeah.
pregnancies, and
Yeah.
I expect, because there's a higher risk of miscarriage, some of these
Yeah.
pregnancies we're not able to then lead you on to testing these children later on in the first place, but also some of them would have been terminated. So that kind of puts
Yeah.
a bottleneck on the
It does,
sample.
it
And how
does.
that influence?
Yeah. So when we started looking at this, we were driven by the question of how can we measure the hormones in utero in a safe way. Obviously, kind of taking a sample direct from the fetus would not be safe, so that was ruled out. Amniocentesis, we think, offers a kind of ethical alternative because the fluid is being collected anyway. So from the research is not adding to any risk, but it does have the limitation you've mentioned, which is that women who have amniocentesis tend to be older. When the test was widely offered, it tended to be for women over 35. And particularly those, and because over 35, your likelihood of having a child with Down syndrome increases, but also other developmental anomalies. So it's not a random or representative sample, but to kind of to measure prenatal hormones, I don't think there's any other kind of way of doing it in an ethical way. I think, actually having said that, it looks like some other methods may be available. So I mentioned one of the studies was looking at meconium, or the first stool that the baby produces, as a reflection of the prenatal hormone environment. And that could obviously be applied to a general population sample, not one that's got various biases. But I think with research, you have to kind of just accept sometimes that there are limitations to how you can generalize.
Maybe baby's hair sample is another option.
Baby's hair sample, yeah, whether it would reflect the prenatal hormone environment or not, I'm not sure. That's what makes this a challenge, is getting in, looking at the hormones at the right point in development, because this is a developmental condition. Thank you very much.
Thank you not only for a model lecture, but for impeccable timing. Can you stay a moment?
Yes, yes.
Professor Baron Cohen will be not rushing away immediately, so if you have questions, thank you very much. I've got to run.
So I mentioned, I think in relation to the last question from the back, if I was explaining the relevance to autistic people, I would see the role of these movements in various medical conditions. That may better screening for certain medical conditions like polycystic ovaries.
I think that's kind of it's kind of more the side.
It is a side, yeah. Because this research is driven by the kind of why does autism happen question. So it's much more basic science. It's not kind of the purpose of the research is not how to help autistic people. That is important. If I was trying to answer that question, I wouldn't be looking at the hormones. In fact, we are interested in that question to look at what would be autistic people's mental health. There, I don't think you turn to things like what kinds of adjustments can be made in society to make you more comfortable for autistic people. So I think this line of research is more kind of basic science rather than.
It seems interesting with a lot of those kind of neurobiology research seems like you can do much of the same with regards to just sex signals in terms of I don't know, the utility of and the issues of findings. I guess just some of the concerns, especially on the research comes from possible applications which they're not.
Yeah.
It's kind of a
I think to sort of answer some of the concerns. Researchers need to be very clear about why they're doing the research and why the reasons they're doing it, but also what they're not doing. So here, for example, we study the prenatal hormones in the state genetics. We're really clear that we're not trying to develop a prenatal test for autism, which is something that many autistic people would have fears about. Why would a test like that be developed? And how might it be used or misused? So it's about kind of transparency, I guess.
Because you're allowed to change.
Go for it.
Yeah.
might be a product of a greater number of neurons.
Yeah,
That has
that's a really good question. Shame you didn't ask it out loud.
No, I was wondering that as well, so I'm glad you asked.
So I think if you've got too many neurons, or just more neurons, you can imagine that the plus side, you might pick up more information from the world. So maybe that helps explain why autistic people notice things that other people miss. Hi.
Turn the kettle on, Simon. Would you
I'm
like
all
a-
right, thanks.
Are you sure?
Yeah, thank you
I
very
just
much.
thought I'd ask because you don't realise it takes a lot out of you, does
It
it?
does. Very sweet.
Okay,
Thank
that's
you.
fine.
But maybe on the downside, too many neurons may result in kind of information overload. And a lot of autistic people, particularly in certain environments, sort of suffer, to use that word, with feeling overwhelmed. That the lights are too bright, the sounds are too loud, it's like too much information. And that could explain why some autistic people need to withdraw. So, in other words, there might be kind of pluses and minuses to the number of neurons.
Yes.
You could also imagine, but this is speculative, that it might affect speed of processing, if you could use that term very generally. Like how quickly can you make a decision or recognize, a situation? It might just take longer if you've got more neurons, So maybe kind of may have some relevance to the disability side of autism.
Yeah. Sorry, can I
Okay.
ask another question?
Go for it.
So just to follow up on my previous one, when you say break the group into cohorts,
Yeah.
do you have any thoughts on how you might go about that and how you would group people, and would that lead to different sort of diagnostic categories potentially?
Right. So I suppose what I had in mind was, like in the study I described, that was a collaboration with Denmark. We looked at babies born in the 1990s, but you could try and repeat the same study with babies born in the 2000s or or before the 1990s. So that way you're kind of looking at, are there cohort effects?
I see. Yeah.
And if autism was being diagnosed differently in one decade to another, that should maybe reveal itself.
It would reveal it, but it wouldn't help research going forward.
Well, you might find that irrespective of the cohort, actually the same patterns appear, and it could be that in certain cohorts, you talk about subgroups, it might be easier to identify whether the findings relate more to, for example, autistic people with or without learning disability.
Yeah.
So could help in that way.
Yeah. Thank you.
Okay.
Thank you for the talk. I was just a little bit I was wondering about when the females with autism, they typically get diagnosed later on in puberty. I wasn't sure because you touched on it briefly about their behaviour and maybe they're masking it, but then I was also thinking about the influx of testosterone in the menstrual cycle, and that can affect inhibitory neurotransmitters and excitatory neurotransmitters. Has that been looked at all, or do you think it's just their behaviours? Because that could also make an effect of why they get diagnosed later.
Absolutely. So what's been done? I think it's been found that girls who are autistic go into puberty a bit later.
Okay.
So if you ask autistic women, when did you have your first period, compared to non-autistic women, I think there's something like a 9-month delay on average. So maybe their early prenatal hormonal differences are having a knock-on effect on menstruation, kind of much later in development.
Yeah, that's why I was thinking. I wasn't thinking like, it can just be them masking it, but
No.
that it's also
So hormones wouldn't just affect behaviour. Could have widespread effects on on the body, but including the menstrual system. Yeah. And you were asking about neurotransmitters, is that right?
Well, there has been some research into like the menstrual cycle affecting GABA and glutamate, but it's very sort of in its infancy at the moment.
Yeah.
So that's what I did with my master's thesis. So
Right.
I was just thinking, could that maybe have something to do with maybe why they get diagnosed later, knock-on effect, that sort of thing. Because I was a bit like, can't just be them covering up. Maybe there's other stuff.
Yeah, that's very interesting. So I don't know whether it's related to why they're diagnosed later, but I think I mentioned at the end of my talk some of the work that's going on in Cambridge where you can administer hormones to, they're called organoids, but they're sort of model systems of brain development where you're using stem cells to program stem cells into becoming neurons, and then you can look to see how the hormones change neuronal development. And some of the hormones are affecting GABAergic, so excitatory neurons particularly.
Okay.
So that could fit with what you're saying.
Yeah. Oh, that's interesting. Yeah, hopefully we can look into it a bit more.
Right.
Autism. Mine was just with, healthy participants with regular menstrual cycles.
Yeah.
But I don't think that's been looked at before. So,
Right. What did you use to look at GABA?
So it was magnetic resonance spectroscopy.
Okay.
So it was the brain scans, but it was MRS, not just the pictures of the brain.
Yeah, brilliant. That sounds really interesting. Is it published?
No, I think it's in multi-site, so it's going to get published,
Oh, okay.
but I don't know exactly when.
Right.
But yeah.
Very interesting.
Thank you. Yeah,
Good.
I really enjoyed your talk, and yeah, it was very
Good.
interesting.
Thank you.
I just had a really quick question on the autistic traits thing. I'll make it quick, but it was
Mm.
about, I was wondering if it's like, if there's any kind of behavioural explanation for, let's say you had like parents with higher autistic traits.
Yeah.
Could there be like a learned behaviour explanation for like children then, instead of it being genetic or biological, it's just like they have higher autistic traits because that's what's been modeled to them in certain things?
I suppose so, in the sense that, all children are kind of influenced by their environment and their parents' behaviour. So modelling must, could well affect autistic kids too. That's interesting. But I suppose the fact that, there have been so far actually all the genetic studies have looked at diagnosed autism rather than autistic traits. So we're